Hello, I'm SA, your AI essayist. Please note that all content, audio and video in this presentation are AI generated. As AI can sometimes produce inaccuracies, I encourage you to verify any factual information presented. With that in mind, let's begin.
In 1962, the scientific community recognized the profound significance of the work on DNA structure by James Watson, Francis Crick, and Maurice Wilkins, an achievement that garnered them their prestigious Nobel Prize in Physiology or Medicine. Their insights revolutionized biology, providing a framework for understanding heredity and laying a foundation for the burgeoning field of molecular biology. This paper served as a culmination of years of investigation, bringing together previous research and groundbreaking experiments to explain the essence of life's blueprint. The work transcended mere description. It unveiled the very essence of biological information storage, transforming our understanding of evolution, disease, and the fundamental processes of life. The intellectual landscape preceding Watson, Crick, and Wilkins' breakthrough was rich, teeming with both progress and enigma. The identification of deoxyribonucleic acid, or DNA, as a primary component of chromosomes had occurred earlier, yet its structure and function remained an impenetrable mystery. Key foundational work by chemists and biologists contributed significantly to this understanding. Owen Chargaff's meticulous analysis of DNA composition provided crucial clues, specifically the observation of the Chargaff's rules, which stated the amount of adenine always approximately equals the amount of thymine, and the amount of guanine always approximately equals the amount of cytosine, within DNA samples across various species. These ratios hinted at a specific structural organization, although the implications at the time were not fully appreciated. Further, Linus Pauling's successful elucidation of the alpha helix structure in proteins, achieved through X-ray diffraction techniques, served as both inspiration and a challenge. It demonstrated the power of this analytical methodology in revealing complex molecular forms and set a standard for structural determination. The race was on to decipher the structure of DNA and unlock its secrets. Watson and Crick, fueled by a mix of ambition, insight, and a spirit of collaboration, embraced the challenge. They utilized the X-ray diffraction data generated by Maurice Wilkins and Rosalind Franklin, the latter whose crucial contributions initially received less recognition, to build a model of DNA's structure. Franklin's meticulous work, in particular, generated high-resolution diffraction patterns, which provided critical details on the molecule's shape and dimensions. The crucial image, photo 51, provided essential information about the helical nature of the DNA molecule. The precise application of physical chemistry and knowledge of bond angles led to the development of their model. The structure they proposed was a double helix, composed of two antiparallel strands, held together by hydrogen bonds between complementary base pairs, adenine pairing with thymine and guanine pairing with cytosine. These base pairs, meticulously arranged in the center of the helix, formed the rungs of a twisted ladder. This organization immediately suggested how genetic information could be encoded, replicated, and passed on to future generations. The proposed model was elegant, intuitively satisfying, and it provided a clear explanation of how the molecule could function as the central repository of genetic information. The immediate impact of Watson and Crick's work was profound, sending ripples throughout biological research. Their model offered a plausible mechanism for DNA replication. The model demonstrated that when the double helix separated, because of the nature of its structure, each strand could serve as a template for the synthesis of a new complementary strand. This semi-conservative replication process immediately provided a basis for understanding how genetic information was duplicated prior to cell division. In addition, the structure instantly explained the nature of mutations, alterations in sequence of bases, which could arise through various processes. For instance, exposure to radiation could lead to changes in the genetic code. This was instrumental in understanding how genetic variation evolved, opening up the field of genetics and driving the understanding of evolution. The model became an indispensable tool for understanding various biological processes, from the simplest bacteria to complex organisms. The discovery catalyzed an expansion in experimental biology as scientists rapidly embarked on investigations to validate, refine, and build upon the influence of the Watson and Crick model on scientific thought and subsequent research has been truly expansive. It offered a foundation for understanding gene expression, the process by which the information encoded in DNA is used to synthesize proteins. Scientists next focused on the process of transcription, the process through which the DNA sequence is copied into ribonucleic acid, or RNA, which then serves as a template for protein synthesis. This work led to the discovery of the genetic code, that is, a set of rules that specify the relationship between the sequence of nucleotides in messenger RNA and the sequence of amino acids in a protein. Deciphering the genetic code accomplished by scientists like Marshall Nirenberg, Hargobind Khorana, and others, represented another monumental step and paved the way for the field of genetic engineering. It provided a means to manipulate and alter the genetic makeup of organisms, the potential for gene therapy, and the development of molecular diagnostics. The Watson and Crick model, through its elegant simplicity, has served as a touchstone for research across a vast spectrum of biology, including evolutionary biology, developmental biology, and disease risk. The initial reception of the model, while celebrated, was not without its complexities. While the scientific community widely recognized the profound implications of the Watson and Crick model, criticism arose, specifically among those who focused on the contributions of Rosalind Franklin. Her experimental data, particularly the crucial X-ray diffraction patterns, were essential in the formulation of the model, yet her participation in the process and her role were initially underplayed, an unfortunate circumstance that has prompted ongoing historical and ethical discussions. Furthermore, because of the relative nascent state of molecular biology and genetics, some of the more detailed experimental validations of the structural model took time to fully materialize. However, even during these early stages, the overarching framework of the model was quickly embraced by the scientific community because it provided a unifying framework for understanding many disparate biological facts. Several key individuals and prior works directly shaped the work of Watson and Crick, exemplifying the collaborative nature of scientific progress. The data gathered by Chargaff regarding base pairing ratios, although unaccompanied by clear structural explanation, provided an essential clue for base pairing. Pauling's successful elucidation of the alpha helix, along with his understanding of molecular structure, served as a blueprint but also as a source of competitive inspiration. In addition, Erwin Schrödinger's work What is Life, published in 1944, posed fundamental questions about the molecular basis of inheritance, and it influenced the scientific community to explore the physical nature of biological information. All of these, in addition to the data provided by Rosalind Franklin and Maurice Wilkins, represent pieces of a larger puzzle that Watson and Crick effectively assembled. The influence of the Watson and Crick model on subsequent scientific endeavours is extensive, and it is evident that the impact is ongoing. Their identification of the double helix propelled research into gene regulation has led to the discovery of regulatory elements, genes, and signalling pathways. It contributed to investigations into the function of different genes, the activation or silencing of genetic codes, and how these processes are disrupted. This has impacted the understanding of diseases like cancer, diabetes, and neurodegenerative disorders. Further, the model also influenced the development of biotechnology. Techniques such as DNA sequencing, polymerase chain reaction, or PCR, and recombinant DNA technology are all direct descendants of insights gained into replicative information storage and genetic manipulation. The legacy of the Watson and Crick model continues to be felt in fields such as genomics, personalized medicine, and synthetic biology. Even today, it serves as a framework for future discoveries. In conclusion, the 1962 Nobel Prize recognized a work that marked a turning point in the history of science. Watson and Crick, in conjunction with Wilkins, transformed our understanding of biology by providing the key to the structure of DNA. It provided a foundation for comprehending inheritance, evolution, and cellular processes. Their work, influenced by the insights from earlier research and the contributions of others, especially Franklin, catalyzed a new era of molecular biology, providing a framework for understanding the essence of life itself. The double helix model has not only provided a foundational tool for scientific research, but it has also, through its impact on areas such as medicine, biotechnology, and evolutionary biology, contributed to the evolution of our entire society. The work continues to inspire scientific innovation.
We really appreciate you tuning into this AI-powered presentation. Our goal is to push the boundaries of AI in content creation, and your input is invaluable. If there's a particular subject you'd like to see us tackle next, or if you have suggestions on how we could fine-tune our AI's creative prompts, please don't hesitate to leave your ideas in the comments section. We're excited to bring you more unique content very soon.